Today we will discuss about glycobiology and how we can relate the glycobiology to develop some of the immune therapies. So, and these immune therapies are based on the glycan epitopes that are found on the disease such as cancer's disease. And we have discussed in our previous videos that in the case of pancreatic cancer, we have seen the many researchers have seen aberrant glycosylation. So this aberrant glycosylation, we expected to find out some glycan epitopes that are unique to the tumor cells only, and then we can use these immune therapy approaches against those glycan epitopes. So this is the initial process to develop this immune therapy. Is first we need to get this glycan antigen in these hard to treat cancer cells, and this once we have identified the unique glycan antigens from the cancerous patients or from the diseased patients, and thereafter we need to develop a drug that can efficiently detect those glycan epitopes and differentiate them from the other glycan epitopes. The more, and in this case the specificity of the drug is the main concern, because non-specific binding may leads to the side effects. So in this case, one of the approach that people used is to first find out the microRNA that produce this kind of glycan epitopes, and then target the drugs towards those microRNA. So this is one of the approach that, so when we target the root cause, that is the microRNA that leads to the generation of those glycan epitopes, then this disruption in the intracellular pathway of the cells the might disrupt the uh, tumor intracellular functioning, and that might affect the survival rates of the tumor. So this is one of the approach. And so after this second step, when we have identified which microRNA leads to this uh, glycan epitopes for glycan epitopes that are found in the case of cancerous patients, then we can target those microRNA. And after that, after targeting those microRNA, we need to develop this is the alternative approach. You can say that we need to develop the glycomarker, uh, or we need to develop the glycomarker that can efficiently target these glycan epitopes. And for this, different lectin studies can be carried out, and that is shown here, that at different stages of the disease, this lectin study has been carried out so that they can effectively target those glycan epitopes. And once they have target the glycan epitopes, they are successful in developing this glyco markers. They can use those glycomarkers in the immunotherapies such as these CAR T cell therapies or immune checkpoint inhibition therapies. These checkpoint inhibition therapies are used to block the receptors that interact with the immune cells and deactivate the immune cells. And whereas these CAR T cell therapies can be used uh, with these glycomarkers, so that these glycomarkers helps in the identification of this aberrant glycan structures on the tumor cells, and then this CAR T cell will trigger the program cell death to those identified tumor cells. So this is how this approach will be worked. However, the future possibilities of the approach is based on the data. That is, once we have enough this glycoinformatics data, we can compare this glycoinformatics data of the healthy patients with the diseased patient, and can try to generate pathways that might cause this disruption, that might cause this aberrant glycosylation in the case of cancerous patient. And then based on this information, we can carry out the experiment on the mouse and try to disrupt those signaling pathways and see what kind of glycan epitopes this mouse will able to generate, and then try to target those glycan epitopes. Once we are successful in replicating this same pancreatic cancer in the mouse models, we can use different immune therapeutic approaches to try to cure those diseases within the mouse, and thereby we will be able to generate glycomarkers that are specific to the antigens that are found in the case of uh, cancerous patients. So in this case, the studies are carried out ex vivo, whereas here the studies are carried out in vivo. So in vivo validations are always better than the ex vivo validations, as uh, in vivo validations uh, conditions, validation, in vivo experimental conditions are close to the actual conditions. So I hope you got an idea how this glycobiology can be used to develop these immune therapies. If you need to understand more about this relation of glycobiology and immunotherapy, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.